Welcome to Influence, the show about one of the university's founding colleges, the College of Agriculture and Life Sciences. I'm your host, Emily Shaw. On today's episode, we're going to be learning more about the Weed Science Graduate Program and some specific research going on in that area. Our first guest is Dr. Paul Singh, an assistant professor in the Department of Plant and Soil Sciences. Welcome, Dr. Singh.
Morning, Emily. Thank you for coming.
Yes, I'm excited to hear more about your weed research, or specifically your weed physiology research.
Yes.
So tell us about that.
Well, all of this started when I was doing my PhD in Arkansas.
Okay.
So I was mostly focused on the weeds and looking at all the molecular mechanisms, the genetics behind all of these competitive traits in weeds.
Okay.
My PhD was mostly in weedy rice. So it's basically rice, but it's a weedy form of rice.
Okay.
So we basically have a lot of rice producers in Arkansas, and Arkansas is kind of the largest rice producing state in the whole US. So what we did was we collected numerous samples from Arkansas, and we actually, numerous samples of weedy rices from Arkansas, and we actually tested and try to see what are the competitive traits in these weedy rices, whether or not they are taller, whether or not these leaves are longer compared to rices, how's the yield, and also we looked at numerous different hull colors, we call it. So, I mean, if people are not aware, weedy rice is actually one of the most troublesome weed in rice. It can cause up to 80% yield reduction in rice, so that's a big thing for the rice producers. Also, in a large field, if you have a large area of weedy rice that's infested, it actually leads to all of that reduction in yield in that whole infestation. So the rice producers really want to get rid of these weedy rices. Now, as a weed scientist, we want to study these weeds instead of getting rid of them. Okay. So what we wanted to see was, what are these competitive traits in these weeds, develop molecular markers for these competitive traits so that the rice breeders can actually use these molecular markers in actually developing a better variety of rice. So for example, weedy rice can actually flower early.
Okay.
So we can actually use this trait and incorporate into rice so that rice can flower early. The growers do not have to wait too long to actually get the yield. Weedy rice can also actually grow taller than rice, so that's another trait. Now, apart from all of this, we also found out that weedy rice has numerous tolerance to abiotic and biotic stresses.
Okay.
And by this, we basically mean that it can tolerate, very high temperatures, very cold temperatures. It can also tolerate drought. So when you don't have any water at all, the weedy rice can still grow.
Wow.
There's also biotic stresses. So by biotic, we basically mean that it can actually tolerate numerous diseases. So numerous fungal, bacterial diseases. So all of these are actually becoming a good resource in weedy rices that we can actually find. So we can actually search for all these traits in weedy rices, and then we can try to see what are the genetic regions or genes that's associated with these traits.
Okay.
And with this, we can actually use this information in rice breeding programs.
Okay.
So that's what we basically be doing, and with that whole information and experience that I got in my PhD, I've been transferring that in this position that I got in Mississippi State University.
Right.
So we've been looking further as to what are these genes involved with these competitive traits. So I have numerous graduate students who's actually working on that. And apart from that, we also looked at numerous herbicide resistance in these weeds.
Okay.
So we have I'm not sure if you're aware of Clearfield rice. So Clearfield rice is basically a variety of rice that's been used in the South mostly by all the growers, most of the growers. So by variety, we basically mean that this rice is resistant to a herbicide called as ALS inhibiting herbicide.
Okay.
And more specifically, it's called as imazethapyr. So that's the herbicide name. So what the rice growers do is they will plant these Clearfield rices and they'll spray them with this imazethapyr herbicide, so it will not affect your rice, but it will go and kill all the other weeds around it. Okay, so this has been a very good technology that the rice producers have been using. And if I'm not wrong, I think this was introduced in 2012. So it's been 2002, sorry. So it's been quite a lot of years already. Now the problem is that growers have started to see weedy rices emerging as well.
Okay.
So they're supposed to be susceptible and they should be killed. But because the growers have been using it for so many years, you're actually having some of these weedy rices evolving resistance to this particular herbicide. So again, as a weed physiologist, we are interested in seeing what the mechanism of resistance is in these weeds. And also, we wanted to find out when did this whole evolution of these weedy rices start? When did they start becoming resistant to this herbicide? So it was introduced in 2002, for example, that whole variety of rice that's resistant to the herbicide, much like how we have Roundup. So it's resistant to glyphosate. So this is resistant to imazethicline herbicide, we call it. So in 2002, it was introduced. After about 5 or 6 years, we started seeing complaints from growers saying that they have weedy rice that's also resistant to this herbicide. So they're not able to completely eliminate this weed in its rice field. So what we actually did was we collected samples before the introduction of this Clearfield rice. So we collected weedy rice samples before this Clearfield rice was introduced. And then we collected weedy rice samples 5 or 10 years after these weedy rice, after the Clearfield rice was introduced. So now we have samples that are before, and we call this historical, and we have samples that are after the introduction of Clearfield. So we did this genetic study, and we found that the samples that were collected after introduction of Clearfield rice had a lot of the genes that was obtained from the Clearfield rice as well as the historical rice. So it basically means that it had a lot of this crossing going on with the Clearfield rice in the field itself, and which is why it tells us why it's actually resistant to this Clearfield rice. So that's what we've been actually doing. And apart from that, a lot of my students in the program, they're actually looking at abiotic stresses, as I mentioned, so cold tolerance, heat tolerance. We're also looking at submergence tolerance. So a lot of these weedy rices can actually tolerate submergence. So it basically means that they are tolerant to flood, for example. So with all of this, just recently, we kind of applied for, one of my students applied for a grant or an application to the NASA. And they are actually more interested in growing rice in, say, for example, Mars. And in Mars, we have different temperatures, varying temperatures, very cold and then very hot. So they became interested in this as well, trying to see how we can actually progress further with this whole project that we have.
Excellent. Well, thank you, Dr. Singh, for sharing that research with us today. When we come back, we'll hear from Dr. Singh's student in the PhD program.
Think you know ag? Think again.
We are creative.
We protect the environment.
We prepare for careers in medicine and conduct research.
We empower people and communities.
We are technology driven.
And we feed and clothe the world. We are the College of Agriculture and Life Sciences at Mississippi State University.
Welcome back to Influence. Our next guest is Chandrea Stallworth, a PhD student in the Plant and Soil Sciences Weed Science concentration. Welcome, Chandrea.
Hi, how are you?
Doing good. So you are under Dr. Singh. He's your major professor.
Yes.
So tell us about your research.
So my research focuses on the abiotic stress, and we are trying to discover weedy rices that are tolerant to heat, cold, and complete submergence, because we use those as weed management tools. So we have a number of screening protocols that we follow. For our cold module, we are exposing our weedy rices to about 10 degrees Celsius weather for extended periods of time to look for those that survive. In our heat, we look at 38 degrees, which is roughly about 100 degrees Fahrenheit, and we do that for about 2 to 3 weeks at a time. And the whole point is that extreme measures to see if we can find anything that escapes, because we know that if they're able to survive repeatedly, there's something genetically going on with these rices that could potentially help rice breeding programs. So we, like I said, we do these in the greenhouse. We grow them in the field. And the whole basis is for a gene discovery project, so that we can hopefully look at the expression in the rices that are currently being planted in the South and across the world, and see if we can actually improve rice breeding. With global warming happening and climate change, we've seen that these extremes have become even more extreme in shorter periods of time. Crop losses have happened at an exponential rate over the last 10 years. And economically, it's beneficial to our farmers for us to be able to discover these genes and help them.
Okay, excellent. So what do you hope to do with this PhD and this weed knowledge and weedy rice research moving forward?
So my hope is to go into R&D research. I have discussed with Dr. Singh that I would like to be an adjunct professor, but I'm really, really interested in gene discovery. I really enjoy my lab work. The rush that I get from being able to see something that, you know, when you're working on a project for months at a time and it's just that one light bulb that goes off, it's elating. It makes you, you know, you kind of, it lights a fire, because you feel like you have done something that only a small portion of the world is able to do. So we have, as he mentioned, we are partnering with NASA. And that would be a research and discovery type of input, where we would be able to actually see if we can grow any type of crops in space, and then going from there, establishing those ecosystems further. So where things are moving in the world, it looks like it would be very beneficial for us to continue this partnership with NASA, and for me to stay in R&D and hopefully continue to discover some genes and crops and be able to overcome a lot of these issues that we're seeing.
Excellent. Well, thank you for sharing with us your research and obviously your passion for your weed science.
Thank you.
Thank you for joining us on this episode of Influence, where we learned a little bit more about the Weed Science Graduate Program and research in that.